Good afternoon. My name is Keay Nakae. I'm a senior healthcare analyst at Chardon. It's my pleasure to introduce our next guest from Catalent, Chief Commercial Officer, Cell and Gene Therapy, Dr. Philip Wells. The format for this session is roughly a 40-minute fireside chat. If any of our audience attendees have questions during the session, feel free to type them into the question box under the video player, and we will try to ask them on your behalf. So Philip, welcome. Could you provide a couple minutes of introductory comments about Catalent for audience attendees new to the company?
Thanks, Keay. Yes, I'm happy to. So, Catalent is a CDMO. Our mission is to develop and manufacture and supply products that help people live better and healthier lives. Our vision is to be the world's most trusted, reliable, and innovative drug development and delivery partner by upholding the highest industry standards and exceeding customer expectations while driving strong, sustained growth for the company. One of our core values is patient first. And we always like to start our presentation with this because it really is fundamental to who we are as a CDMO. And what patient first means to us is that every decision that we make, everything that we take on behalf of Catalent, we always ask the question, what is the impact on the patient? And we use that to drive all of our decisions. And we have found that as long as we stay true to that, we always end up with the right decision. In terms of the focus technology-wise with respect to Catalent, we're very much focused on solving complex manufacturing problems. The company was originally founded on the soft gel technology, but everything that we do is fundamentally to, complex manufacturing and how to deliver products, whether it be the soft gels, whether it be fast dissolving tablets, gene therapy, all of these, fit that mold of technologies that are, complex and require, a deep expertise and knowledge. Catalent is broken up into several different business units, those that are focused on oral, those that are focused on biologics. There's a portion of the company that is focused on clinical supply, getting materials to the clinics, and then there's the cell and gene therapy portion of the company, which I am a part of. So Catalent is a global CDMO. We have sites around the world. You can see in the green colored dots the locations for the cell and gene therapy portions of the company. So we have sites in Maryland, sites in Houston, and sites in Gosselies, Belgium. So Catalent Cell and Gene Therapy, as mentioned before, is basically a combination of several different companies. So Catalent itself has deep expertise in biologic manufacturing as well as the clinical supply services. They then added the gene therapy expertise with the acquisition of Paragon, which occurred in May of 2019. And then about a year ago, Catalent acquired MasterCell to give us the cell therapy manufacturing capabilities. And then just most recently, we acquired Delphi to provide plasmid manufacturing. So we have an integrated solution, from the plasmids to the viral vectors to the cell therapy, and then to the storage and distribution throughout the world. In terms of our different facilities, as I kind of mentioned earlier, we have some that are in the US and some that are in Europe. For plasma manufacturing, we actually have two sites, one, the Delphi facility that's in Gosselies, Belgium, as well as a site in Rockville, Maryland. It was a site that is dedicated to microbial and plasma manufacturing. For the viral vectors, we have a couple of facilities that are focused on phase 1-2 clinical manufacturing, as well as the development of the manufacturing processes for them. One facility is in Gaithersburg, the other is in downtown Baltimore. And then we have the phase 3 commercial facility, which is located near the Baltimore-Washington International Airport, hence the BWI campus designation. That's 2 buildings, over 350,000 square feet of manufacturing. And actually the building number 1 is the first FDA-approved commercial CDMO manufacturing facility for an AAV gene therapy product. And then for cell therapy, we have the sites in Gosselies, both clinical manufacturing as well as future plans for commercial manufacturing. And then in Houston, we have our site to do additional clinical manufacturing. And with that, I will end my presentation and happy to answer any questions.
Well, great. Well, let's start at the top and kind of work our way down. I think our attendees are well aware of the explosive growth in cell and gene therapy, all the investment dollars going into those emerging companies, all of the products in their pipelines, and all of this has led to a situation where demand currently exceeds manufacturing capacity supply. Compounding the capacity constraints, the manufacturing of the CGT products is complex, and many have stated or would use the analogy that the current need for manufacturing innovation is similar to the evolution that occurred with the introduction of monoclonal antibodies, which themselves were much more complex than small molecules at the time. Ultimately, fundamental principles that underpin the success of monoclonal antibody manufacturing innovation was a solid understanding of the product biology and its critical quality attributes, then designing processes to control for these, and then planning for analytical studies and tech transfer required to advance these next generation processes into the clinic and then commercially. So in your view, Philip, where is the industry in its understanding of the biology of CGTs, given the diversity and variety of, autologous, allogeneic cells, AAV, lenti, adenoviral vectors?
Yeah, I think it's we've come a long way, but there still is a long, many iterations, next generations left to see. When gene therapy first or cell therapy first came in, it was very much based on processes that came from academic facilities, labs. And so it really wasn't industry standard. So, the first wave of changes to the manufacturing processes, for these types of products was to basically incorporate a lot of the principles that we are using today for antibodies and proteins. So getting, into suspension cultures, getting into bioreactors, getting into, column chromatography, filtration, rather than ultracentrifugation. And so a lot of that has occurred, and it has, made a lot of a big difference. We use a lot of the, again, also similarly on the analytical side, using a lot of the same principles, host cell protein, host cell DNA, all those things that were used and understood from the protein antibody world was incorporated into these processes. But, we're now at a stage where we have to, look at what's different about these types of products and develop processes and systems that are unique there. And so that's where we currently stand. There's been some implementation, but there still is a lot more room to grow here into improving, the upstream, the downstream, and the analytics for these types of products.
So given this complexity, the CDMO model is very much at the heart of CGT manufacturing and compared to traditional biologics, in terms of allowing these emerging companies to drive their products forward beyond the acquisitions that you mentioned. And beneath that, how has Catalent shifted its business model to support the needs of these small biotechs focused on CGT?
Yeah, I mean, it's a different approach. I mean, I think in cell and gene therapy, the bulk of the product sponsors are small biotech, and they take a different type of relationship than you would see from a larger pharma company or biotech company. They are oftentimes limited with respect to prior knowledge on how to manufacture these at scale. So they really are relying on their CDMOs to provide that solution. But they also have different needs with respect to they have an urgency to their corporate mission that you don't see from the pharma companies, and they need a partner who really understands and embraces that urgency and how important it is to get these products because they are oftentimes treating diseases for which there isn't a current therapy, and for which there is oftentimes some morbidity that's associated with them, to getting these treatments out there and treating these patients and hopefully finding a cure. And that is a different approach that needs to be had, and it's very much focused on a partnering mentality.
And so in terms of working with these companies as they mature and they go through different stages, talk about the flexibility you offer as a CDMO, whether it's maybe early on fee for service, all the way up to a dedicated line, and even the nature of your contracts, whether they be take or pay or what have you.
Yeah, I think you hit on a really key word there, is flexibility. That is definitely something that the small biotech companies need in a partner. They need the understanding that it's not cookie cutter, it's not one solution fits all. Each of them needs a partner who's willing to listen to what they need and to offer solutions, and oftentimes they're very tailored solutions to their particular program. So it is oftentimes a batch contract where they pay for a certain number of batches. But as mentioned before, there's also this need for potentially dedicated capacity. They do have a deep pipeline. They do have a lot of products that they will need to be manufactured, and with the capacity constraints that are out there, they want to really be sure that is secured. So some of them will want to enter into a long-term arrangement where they are guaranteed capacity, and it could be something that is very specific, that they get a suite and it's theirs for the next foreseeable period of time, or it could be something where they get a certain number of slots per year that are guaranteed to them. But all in all, it really is about providing them with flexibility and providing a true partnership in developing these types of processes. But as they evolve, their demands, their needs change, and we have to evolve with them.
And just expanding on that point, as they grow and evolve, some of them may choose to move to more of a hybrid strategy. So if they choose to do that, in your experience, just from a risk-benefit perspective, which tasks should they be able to take on and optimize that risk benefit, and which are maybe better suited for the CDMO to continue to do?
Well, I would think, again, each situation is always a little bit different. But oftentimes, what they're coming with is a limited expertise and knowledge about how to manufacture this at scale, how to get regulatory approval. And so I think, at least in the initial point in time, they really need to rely on their trusted CDMO partner to provide that guidance and provide that expertise and knowledge. But at the end of the day, the CMC for their product really is kind of their product, and they really need to own that. And by to own that, I mean they need to understand it fully. And so I think it's very important, that if they really want to do this well, that they need to invest in some internal development, manufacturing capability for them to leverage our knowledge and our expertise and understand their process, understand, the critical quality attributes that are associated with their product, and not to rely on a CDMO, even if they are the best, most trusted partner for that. Because at the end of the day, they're the sponsor and they need to own their product. I think, having some manufacturing capabilities, has some benefits, right? You have a second source, guaranteeing supply, but of course you're investing a lot of money, into those facilities, into that infrastructure. Starting up a facility, for the first time is never easy. Done it a couple of times, and there's always challenges. And that's with the benefit of past experiences. So, I would say, consider wisely whether to build, any internal manufacturing. But at the end of the day, you're never going to build enough manufacturing capacity to handle the best case scenario for your product. It just wouldn't make sense. If you're going to build some capacity, you should always build conservatively and then use a CDMO to supplement, that capacity.
Okay, great. Over the course of the last 2 days, we've heard a lot about different stages and aspects of current manufacturing that certainly could benefit from further improvements. So for some of the following, can you talk about what Catalent is doing to address these areas? Let's start first upstream, the need for more stable cell lines.
Yeah, I mean, we're investing in developing those types of technologies. We are also, very active in surveying what is out there in terms of either getting access to or securing licenses to next generation technologies for the production of these types of products. And I do agree that, a stable cell line, it will be very transformative in this particular field. So, we have some partnerships that I can't divulge at this point in time, but we're very active in this field, and it's very important.
Okay, how about better analytical characterization? So, better assays to assess things like potency and consistency.
Yeah, I mean, we've invested a lot already in our analytics. We have, the full standard gamut, but, things such as, AUC, mass spec, these things are less common in the CDMO world, more likely to find those at a CTO. So, we have found that having that expansive analytics really does serve our customers well. So, just an example for AUC, when we first start developing an AAV manufacturing process, you always look for yield. PCR is the most common way to determine, what your yields are. We also do infectious particles, a TC-ID50 assay. That's something that's kind of unique that we implement very early on in the process. But there is the issue of the packaging not being optimal, and you really best see that by doing AUC. So having analytical ultracentrifugation, on site, we actually have it at 2 different locations, and to be able to perform that type of analysis very early on, before you've invested a lot of time and effort, to make sure that the vectors are packaging properly and you're getting fully formed particles, I think is invaluable. So yes, analytics is a key and will be even more so, I'm sure, moving forward.
Okay, how about improved purification methods?
Yeah, I mean, I think anything to improve yields, whether it be to produce more or to recover more, is highly beneficial. I mean, I think we're getting pretty good recoveries right now in the 40% range. So they're still leaving 60%, that is material that is not being used, that can be, obviously improved upon. But we're in a fairly good situation. But again, the other thing to think about is not just the performance. I think everything has been driven by performance up till now, which has led to a lot of these improvements. But we also need to start thinking about cost. So trying to find ways to get similar performance at a reduced cost.
Right. And then how about improved QC to facilitate shorter product release times?
I mean, that's essential. I mean, you hear about the COVID vaccine, right? And these things are manufactured, but what's holding up delivery, and it's all this release testing and making sure that the product is suitable for use. So ways that you can find to decrease that testing timeline, which can sometimes be 3 months to get a testing result back. For example, adventitious agent testing on your viral vector product to make sure that it's not contaminated. That takes 3 months to get back, from these CTOs. So finding ways to improve, coming up with solutions that can do this faster, but still not put the product at risk, are very, very important. And so, we're investing, one way is to try to bring some of these assays in-house so that, we then have more control and are able to expedite as much as possible.
Okay. In terms of new methods, new processes, in your position as a CDMO, are you seeing any notable shifts in things such as moving from adherent to suspension or use of baculovirus cell lines?
I would say there definitely has been a shift, from 5-6 years ago, where everything was very much adherent based, to now where it is very rare to see someone coming in wanting to manufacture adherently and not in suspension cells. So there definitely is a move to suspension. I would say transient transfection of mammalian cells still really is the predominant way to manufacture these products. For some of these indications where they really need just such large amount of material that they just can't think of a way to do it transiently, they are moving to baculovirus. But there has been a lot of improvements on the transient manufacturing process, so that it's more of a viable option for a lot of these types of products. I mean, say 4 years ago, getting 10 to the 10th viral genomes per mil of production was very much the norm. But now we're a log better, pushing 10 to 11, mid 10 to 11 viral genomes per mil from a transient manufacturing process. So there certainly is a lot of improvements that have enabled transient transfection to still stay the predominant way to manufacture.
What other innovative technologies do you see coming to the forefront in the short or mid-term?
Well, I mean, I think you mentioned the predominant one, which is engineered cell lines, finding a way to improve yields by making the production cell better in producing the products. That is going to be transformative, and it's really, hopefully not too far off in the future. I think the other thing is moving towards a lot more automation to manufacture. So right now, a lot of the manufacture of these types of complex biologics, these advanced therapies, is very labor intensive, operator intensive. A lot of interventions, manipulations, and each one of those runs the risk of potential contamination, right? So finding a ways to make this more automated, more closed system, avoiding the potential for any in negative impact due to the operators, I think is something that where the industry is moving towards.
Well, with automation, but even without it, I guess enhanced use of informatics to facilitate things like electronic batch records and electronic submission can certainly provide cost and time advantages. So where's Catalent at its facilities in terms of implementing these types of technologies?
Yeah, I mean, we're very much making a big push to bring more electronic systems to our facilities, especially our phase 3 commercial facilities. So digitizing batch records, getting all the data that is collected at each of the individual silo pieces of equipment, and getting that more pulled together into a single database, so that you can very quickly and easily look at the characteristics of the manufacturing, to understand how things are performing, see if things are starting to change and to move off trending, understand the trending, and to really get on top of this manufacturing, because it's not like we have a long history here with manufacturing these types of products that we know how it's all going to play out. I mean, a lot of this is evolving, and so we need to really be keep our eye on these types of trends that may be indicative of potential areas that we need to investigate.
What you do doesn't occur in a vacuum. You gotta, what you do, the regulatory agencies like the FDA, EMA, and others have to be comfortable with. They have to understand it. So from your perspective, how up to speed are they with not maybe what you do now, but what you're, hoping to implement in the near future? And, what do they still need to get comfortable with, where the state of manufacturing is?
To be honest with you, I think the agencies have been really, really supportive. I mean, I think they embrace the concept that these types of therapies are new and different, and so they're not coming in narrow-minded. They're willing to consider. They're letting science speak as to what makes sense, and because these are life-changing, and again, oftentimes for terminal indications. They're willing to think outside the box a little bit. With that said, as these products mature, as they learn more, as different parties come up with ideas that make the manufacturing the products better, and they become visible to them, they start to make strong recommendations that those similar types of things are incorporated elsewhere across the board. So, as they understand certain tests, as they understand, certain concepts with respect to, say, like 293 cells and 293 T cells, they implement, procedures that and recommendations that improve the product. But to be completely, the agencies have been very supportive of these particular products.
Okay. Well, let me ask you a question that we've gotten from one of our audience members. Since Catalent Paragon has well experienced producing clinical grade AAV vectors and virus-like particles using both HEC-293 and SF9 respectively, can you elaborate in brief the pros and cons of each platform for AAV manufacturing from a CDMO perspective?
Wow, that sounds like almost like a softball question here, a pitch here. So my apologies if that is the case. But the transient transfection system, I think, has the advantage that a lot more products have been produced in that, and it's using a cell line that is most similar to its native host to manufacture these products. So there is less risk that the product itself, would be vastly different from a wild type AAV. It also provides some increased speed. So once you have plasmids at hand, it's very, very seamless to kind of scale up your cells, transvect your cultures, and produce the AAV particles that particular way. Whereas in the baculovirus, you'll need to make master viral banks for both the gene of interest as well as for the capsid and release those. And so that does take additional time. The advantage of the baculovirus system is the fact that, because it's not using the transient process, which is basically you're taking the plasmid DNA and you're complexing it with a chemical, and that by forming together these little particulates, it gets incorporated into the cell. But those particulates, are very unstable. So that's part of the trick, is to be able to make that complex of DNA and transfection reagent, get it into the bioreactor in a sufficient period of time for the transfection to be optimal. That, A, limits scale, and B, it also creates some variability, based upon the timing of doing those steps. The virus addition, using the baculovirus, does create the ability to go to much higher scales. So you're not, limited to, 500 or 1,000 liter, whatever is the current limit of transient transfection. You, technically speaking, you go to 30,000, 40,000 liter tanks, because what you do is you grow up the cells all the way up, and then you add the virus into it. So there really isn't any time requirements or dependency with that particular processing step. So the SF9, much greater scale. It also tends to have at least a little bit higher yields as compared to the transient. We also do see a much higher full to empty ratio from the baculovirus right off of the bioreactor. Of course, that can be fixed, with downstream processing, but it does make your downstream, a little bit easier if your ratio is much more towards full than not.
Great. Philip, the development of the available COVID-19 vaccines has been accomplished at unprecedented speed. Besides a large amount of money that was thrown at the problem, what do you believe are the key things learned that can be leveraged going forward?
Well, I think, it certainly has shown that when the agency and the drug developers and the manufacturers work together, things can happen, at a much faster pace. I think we started to see this, several years ago with the Ebola crisis, the agency being a little bit more accommodating, proactive, and that helped, I think, set us up where we're, today able to move so fast on this COVID vaccine. It also helps that, there was quite a bit of investment in establishing technology platforms that could react quickly to this. So, whether it be the mRNA, I think part of the funding for it came from the concept that this would be a way to make vaccines, a lot faster than a traditional mechanism. Similarly, you have Novavax with their virus-like particle that very much was funded by a lot of government, BARDA funding, DARPA, and such like that, which was, seeing the possibilities of using this as a platform that could very quickly react to the situation in the world and create a therapeutic. But what I would say the other, I think, from a manufacturer, what we see that has a lasting impact is really, the COVID situation really has affected raw materials and supplies. Getting those in sufficient supply to manufacture has been a challenge. If you're not manufacturing COVID vaccine, if you're manufacturing for some other therapeutic, even if it is a life-threatening or life-saving therapy, getting those materials, getting that testing done can be a challenge. And so being very proactive about, having second sources, maybe even third sources, and constantly checking with those sources about their, availability, about their ability to deliver, are they impacted with rated orders, things like that. We've created a whole war room within our commercial manufacturing facility really track all of that across all of our different customers to make sure that none of the batches are impacted. And it has been a big challenge, but a lesson learned that, you need to plan for all the different contingencies because you never know what may happen.
Great. A lack of qualified workforce or talent has been cited as a current capacity constraint multiple times over the last couple of days. What is Catalent's strategy for attracting, developing, and maybe most important, retaining high quality people?
Yeah, I mean, when you talk about the capacity crunch, it is very much about the skilled workforce, people that have the understanding on how to make these products rather than just having a facility. I mean, anyone can build a box, but finding skilled labor is the big challenge. So we spend a lot of time and effort in recruiting people and then creating a workplace where they want to come to work. The fact is that we get to work on some very interesting technologies, and it's for very important therapeutics, whether it be vaccines that can get the world back to business as usual, or for gene and cell therapies that are treating orphan rare diseases for which there are no treatments. It's very much keeping the eye on that, as well as ability to learn a lot of different things. I think that's one of the advantages at working at a CDMO as compared to a biotech company, is that you get exposed to a whole variety of different technologies. And so someone could spend some time with the viral vectors and learning all about AAV and lentivirus and adenoviral-based vaccines, and then can do a rotation at a cell therapy facility and learn how to manufacture or develop those types of products. So the ability to be exposed to a variety of different types of products, I think, is invaluable for someone's resume, for someone's future employment. So it's having a lot of training, a lot of room for advancement, and then to work on cutting edge technologies.
Okay, well, let me ask you to look forward for me. 5 years from now, what do you think will be the biggest changes in the manufacturing landscape?
Yeah, I mean, I think we will continue to see improved yields. I think we will see a big driver in trying to bring down the cost of goods. So finding those paradigm shifting technologies that allow to not only manufacture at larger scale, but to do it more cost effectively. As we talked before, I think it's a lot more automation, a lot more integrating of data to really fully understand your process and understand your product, and it's then taking what we've done thus far and treating things that we even didn't think of. Right? I mean, I think that's the great thing about science, is that soon as a technology is out there are really smart people thinking about how else to use it to do something completely different. So I would be surprised if we're just doing monogenic diseases with AAV 5-10 years from now. My guess is that we're going to start treating a lot of other more complicated diseases and using that technology platform for that purpose.
Okay, one last question, Philip. So let's summarize all of this. And how does Catalent differentiate itself to stand out as the preferred CDMO partner?
I mean, there's a lot of things. I mean, I think the most important thing is that, we very much view ourselves as a true partner with our clients. And so, we bring a lot to the table, that is, past experience and expertise, knowledge of what's worked in the past, what doesn't work, what are the potholes that need to be avoided. I often talk about when, we're looking at, potential opportunities, the type of customer that we want to work with, and that is a customer that's, with a strong management team that's already developed a drug previously, been there and done that, and understands, how to avoid those potholes in developing those drugs, having someone to steer the ship from the customer side, that can do that makes our lives a lot easier. And similarly, I think picking a CDMO that has the past experience in developing these processes, manufacturing them for the clinic, manufacturing them commercially, knowing what is going to be needed as you transition from these different stages, especially since these transitions are going to occur so fast, right? It's not your normal typical paradigm where you have a stage, then you get data, and then you assess, you try to come up with a strategy, you do your next step. I mean, here it's just a sprint from the beginning to the end. So finding someone who can get you into a commercially viable manufacturing process right from phase one, and to be able to optimize it, further develop it, do the analytics, give the regulatory support. I think that's invaluable. And I think that's, where we separate ourselves. We provide all of that. We provide, in one roof, the ability to manufacture the plasmids, the viral vectors, the cell therapies, regulatory supports, supply and storage, having that complete solution.
Okay, well, very good. So with that, we've reached the end of this session, and we've reached the end of this year's Manufacturing Summit. So Philip, thank you for being with us, and I want to thank all of the audience participants who've joined us over the last 2 days, as well as all of our presenters. So with that, we'll call it a close.
Well, thank you very much for having me. It was a pleasure.
Great, thank you.